Hi guys, welcome back to Caribbean Reach Academy's YouTube channel. I am Rachel Harris, and in today's video, we are going to be conducting an experiment. In the previous video, we spoke about why plant leaves change their colors. In today's video, we are going to be looking at how to separate these pigments using the chromatography separation technique. If you are new to this channel, don't forget to go ahead and click that subscribe button. Click on the notification bell so you don't miss when we post our new videos. Come along! To start the experiment, we will need a blender to grind the leaves. We will also need the leaves, of course, which we will extract the pigments from, and we will need alcohol to help with the separation of those pigments. We also will need hot water to speed up that process. All of this is necessary for this experiment, as we would love to see how chromatography works with biology. Let's begin. Here we are tearing the leaves into bits and pieces so that we can actually have more surface area and room for extraction. So we will simply tear the leaves, pour the alcohol on the leaves, then we go ahead and blend. As soon as we blend and have extracted a suitable amount of the chlorophyll, then we can simply go ahead and pour it into a glass, place in a hot water bath to speed up that process. We let it sit for a few minutes. Once we have let it sit for a few minutes, we then get our paper towel or filter paper or chromatography paper and now leave that for roughly 30 minutes, and then we can see the results with the two bandwidths with the chlorophyll and the carotenoid pigment. Thanks for watching, guys. I really hope you were able to see these pigments. You can go ahead and try this experiment. It's pretty easy and simple. So don't forget to like, comment, share, and subscribe. Turn on your notifications so that you won't miss when we post our new videos. Have a good one.